Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. In this video, I want to presentation about biotechnology enzyme by Group 12 on behalf of Rahmawati 2001 075 11011. Biotechnology enzyme. Biotechnology itself is the application of science and technology principles to the processing of a material by involving the activities of living bodies to produce goods and or services. Biotechnology in the modern era now produces many products on an industrial scale. In adolescent biological agents, biotechnology plays an important role for enzymes, so that enzymes play an important role in industry. Definition of enzyme. Enzymes are non-toxic proteins but are able to accelerate the rate of semi-cell reactions in mild temperatures and degrees of acidity. The products are so specific that they can be calculated easily. Various enzymes that are used commercially are derived from plant, animal, and selected microorganisms. Enzymes traditionally obtained from plants include proteases, fipronficin, and bromelain, amylase, lipoxygenase, and curtain specialized enzymes. From animal tissue, the enzymes are pasteuritic, trypsin, lipases, and enzymes for the manufacture of butter. From animal tissue, the enzymes mainly are pancreatic trypsin, lipase, and enzymes for the manufacture of butter. From the two sources of plants and animals, many problems may arise normally. For enzymes derived from plants, problems that arise include seasonal presence of concentrations and high processing costs. Weekly planner. Industrial enzymes production currently relies heavily on the deep fermentation method. The use of microorganisms as a source of enzymes production was developed for several important reasons, namely: number 1, normally has a high specific activity for only the week of the product. Number 2, seasonal fluctuation of raw material and possible plant shortages due to climate change. Number 3, microbes have a wide range of characteristics such as pH, caloric, and temperature resistance. Number 4, the genetic industry is increasing so much that it is possible to optimize the yield and type of enzymes strain selection, mutation, induction, and selection of growth conditions currently using advanced gene transfer technology. Some of the enzymes used on an industry scale are extracurricular enzymes. Enzymes that are normally produced by microorganisms according to their substrate in the external environment and are analogous to this digestive enzymes in humans and animals. At the end of the fermentation process, the ideal conditions are a liquid with a high enzyme concentration, a biomass organisms that is easily separated. Enzyme application. Thousands of years ago, processes such as brewing, bread making, and cheese production involved unknown enzymes. In this conventional manner, the technology in interested with enzyme conversion before a coherent canalite building is developed. Enzyme structure: A. Whole enzyme. Enzyme with a perfect structure and can catalyze enzymatic reactions. B. Active cancer. Part of the enzyme where it reacts with the substance. C. Protein section of enzyme. And D. Non-protein parts cofactor. Classification of enzyme: A. Oxidase electron transfer. B. Transferase transfer of functional. C. Hydrolysis reaction. D. Lysis addition or elimination reaction. E. Isomerase removal of inner group molecule. If ligase bond formation, C is COCN. Several enzymes acted in the technology recombinant DNA. Nucleic acid with breaking the phosphodiester bonds. Ligase connect nucleic acid DNA ligase polymerase makes a copy of a molecule DNA polymerase, and topoisomerase makes or changes DNA binding nuclease. Thanks for attention. Assalamualaikum warahmatullahi wabarakatuh.